My name is Lauren Savetti, and I'll be discussing the gut microbiome lab. The gut microbiome in general is the different types of bacteria inside the body's gut. It is closely related to health in the body. One physiological condition that is related to the gut microbiome is type 2 diabetes. These two are related because type 2 diabetes is a disease that causes an imbalance in blood glucose levels. The gut microbiome may reshape intestinal barrier functions and host metabolic and signaling pathways. This can directly or indirectly cause the rapid progression of insulin resistance in the body. There are thousands of different microbes in the gut. With certain dietary factors, it can strongly affect the gut microbiome composition and promote dysbiosis. Their relationship is correlative. Another physiological condition that is related to the gut microbiome is autism. These two are related because of the brain-gut link. A study was ran on rats that did not show signs of autism, and when they were introduced to feces from humans with autism, they responded by showing signs of autism. While this does not prove that the bacteria in the gut can cause autism, it does show that makeup of the gut can contribute to hallmark features of this disorder. The way this changes behavior is since the brain and the gut are connected, it changes the way the brain makes different proteins. Their relationship is correlative. Our first round of animals was 4 dogs, 2 of which who were healthy and 2 who were not. The first was a 12-year, 11-month-old female border collie corgi blue heeler mix of poor health who was obese with cancer. The next unhealthy dog sample was a 9-year-old male beagle of poor health and was obese. In comparison, a 6-year-old male shepherd hound mix of very good health and no health issues was used, as well as a 12-year-old female West Highland Terrier of very good health. Protocol following the BIOL200 lab manual was used to complete this experiment. The first step was the purification of DNA from fecal matter using a QIAGEN kit, according to the manufacturer's instructions. The next step was quantification of DNA in the samples, as followed by qPCR setup and amplification of the DNA. For the purification of the DNA from fecal matter, we weighed out 0.2 grams of the samples into 2 milliliter centrifuge tubes and placed them on ice to prevent degradation of DNA. Next, we added 1 milliliter of Inhibit EX buffer to each sample and vortexed until thoroughly homogenized. Inhibit X removes inhibitory substances by separating them from the stool samples. We then conducted heat suspension for 5 minutes at 70 degrees Celsius, vortexed the samples for 15 seconds after 2 minutes, then continued heating for the remaining 3 minutes. The heat lyses bacteria cells in stool at 70 degrees Celsius. Next, we centrifuged the pellet stool particles, then pipetted 500 microliters of the supernatant in a microcentrifuge tube with 1.5 milliliters of proteinase K, an enzyme that cleaves peptide bonds. We then added 500 microliters of buffer AL, a reaction buffer for proteinase K. Following this, we incubated at 70 degrees Celsius for 10 minutes. 500 microliters of 96 to 100% ethanol was added to lysate, and we vortexed the sample to allow binding of DNA to the spin column. We then transferred 750 microliters of the lysate to spin column and centrifuged for 1 minute at 14,000 RPMs. Then we had to discard contaminating solutes that were in the liquid that flowed through. We opened the spin column and washed with 500 microliters of AW2, which removes impurities, centrifuged for 3 minutes at 14,000 RPMs twice, and discarded the filtrate each time. We then transferred the spin column into a new microcentrifuge tube and added 200 microliters of buffer ATE onto the spin column membrane. This disrupts DNA interaction with the membrane and allows DNA to flow through the buffer. Finally, we incubated at 1 minute at room temperature and centrifuged for 1 minute to elute DNA. Next was the quantitation of DNA via the NanoDrop spectrophotometer. We utilized the NanoDrop 2000 software for nucleic acid. DNA absorbs light at 260 nanometers and protein absorbs it at 280 nanometers. We use this information to calculate the concentration of DNA and to determine the ratio of absorbance at 260 nanometers to 280 nanometers, which is a measurement of purity. We loaded 1 microliter of DI water onto the lower pedestal, lowered the arm, and performed a routine verification. We also wiped the upper and lower pedestal with a Kim wipe between each measurement and changed the micropipette tips between each sample. We loaded 1 microliter of DNA elution buffer onto the lower pedestal, lowered the arm, and measured the blank. For each sample, we loaded 1 microliter of the sample onto the lower pedestal, lowered the arm, and measured the concentration and the 260 to 280 nanometer ratio. Last was the normalization of DNA samples via qPCR. For each of the samples, 5 microliters of 2x master mix, 1 microliter of sample DNA, 2 microliters of nuclease-free water, 1 microliter of forward primer, and 1 microliter of reverse primer was used. The negative control contained all measurements except extracted DNA. The positive control also did not contain extracted DNA, but did contain control DNA. The forward and reverse primers bind to the DNA and allow for DNA polymerase to bind and begin synthesizing DNA. This round was with bacterial and fungal primers. SYBR green binds to double-strand DNA and emits fluorescent light, which correlates with amplified DNA, thus giving initial sample amounts. The positive control contained template DNA in case other reactions produced no product, which would tell us that something went wrong during protocol or DNA extraction. The negative control tested for cross-contamination. Water was added in place of template DNA, and no product should be seen in the reaction. The results for our spectrophotometry showed the different absorbance ratios and concentrations for each sample. Our first unhealthy sample had an absorbance at 0.95, a ratio of 1.43, and a concentration of 47.5 nanograms per microliter. Our second unhealthy sample had an absorbance of 0.042, a ratio of 2, and a concentration of 2.1 nanograms per microliter. Our first healthy sample had an absorbance of 0.082, a ratio of 2.69, and a concentration of 4.1 nanograms per microliter. Our second healthy sample had an absorbance of 0.105, a ratio of 1.66, and a concentration of 5.2 nanograms per microliter. The CQ values that were gathered from qPCR indicate if there is DNA present. A value of less than 15 shows a very abundant amount of DNA. 15 to 20 is the target range, and 20 to 25 is a rare range. CQ values above 25 show that the target DNA is absent. After our first round of qPCR, our positive control had a bacterial CQ value of 13.43 and a fungal CQ value of 31.71. Our negative control had a bacterial CQ value of 27.67 and a fungal CQ value of 34.08. The positive control showed that a good amount of DNA was present, and a negative control showed no contamination. Our healthy samples had bacterial CQ values of 26.43 and 26.33, and fungal CQ values of 35.42 and 32.35. Our unhealthy samples had bacterial CQ values of 22.0 and 25.28, but only one of them showed a fungal CQ value of 33.55. For our second round of qPCR, we changed our question, hypothesis, and samples due to lack of present DNA from our first round samples. Our new question asked if the age of dogs affects their gut microbiome, and we hypothesized that if the dog is younger, then there will be more bacteria present in the gut microbiome. We used new samples for the second and third rounds of qPCR. One of our young dogs was a 4-year-old female pit bull of very good health, and the other was a 5-year-old male pug with average health. For our older samples, we used a 10-year-old female boxer of average health and an 8-year-old female boxer with very good health. The ages were very different and allowed us to view a comparison, and the health status of all the dogs were relatively similar. For the phylo testing, we chose to test for acetobacteria and firmicutes. We then conducted the same normalization of DNA procedure from our last trial, but without a positive and negative control, as they had been values that fit our needs. After our second round of qPCR, our young samples had acetobacteria CQ levels of 32.92 and 31.49, and our firmicute test had CQ values of 24.46 and 23.37. These results had ratios of 2.835 times 10 to the negative 3 and 3.586 times 10 to the negative 3, respectively. Our older samples had acetobacteria CQ levels of 30.77 and 29.73, and the firmicute CQ values were 24.03 and 25.02. The calculated ratios were 9.356 times 10 to the negative 3 and 3.819 times 10 to the negative 2. Though the values did not hit a very good target range, the DNA test shows that firmicutes are present in the samples we took. The next round of qPCR tested for actinobacteria and Bacteroidetes. The procedure was conducted the same as previously stated. After our third round of qPCR, our young samples had actinobacteria CQ values of 27.13 and 29.22, and our Bacteroidetes test had CQ values of 30.54 and 26.49. These results had ratios of 0.10657 and 0.15094, respectively. Our older samples had actinobacteria CQ levels of 29.17 and 29.16, and the Bacteroidetes CQ values were 30.09 and 26.47. The calculated ratios were 0.5290 and 0.65060. Both of these samples did not have CQ values low enough to indicate that these phyla were present in our DNA. Extracting the DNA involves measuring the absorbance, ratio, and concentration. A concentration of greater than 1 identifies high enough concentrations to be able to dilute the solutions, and all of the concentrations recorded were greater than 1. The ratio helps us identify purity among the DNA, which means absence of RNA. Samples 458 and 318 in the first round had ratios between 1.5 and 2, therefore had a greater purity among the 4 samples and less RNA present. The negative control in the first round had a bacterial CQ of 27.67 and a fungal CQ of 34.08. These numbers show no DNA to be found, which is accurate due to no addition of extracted or controlled DNA. The positive control in the first round of qPCR had a bacterial CQ value of 13.43 and fungal CQ value of 31.71. Both of these numbers should have been under 25 due to the addition of controlled DNA and knowing the exact value that should be present. We did not conduct a positive or negative control for rounds 2 and 3. The negative controls in round 2 and 3 of qPCR were all values well over 25, as stated before that it is expected due to no addition of DNA. Differences among the gut microbiomes did not vary as much as we would have liked. Our 2 younger dogs had bacteria present in their microbiome like we expected, but so did one of our older dogs. The younger dogs had CQ values of 24.46 and 23.37, whereas the older dogs had values of 24.03 and 25.02. With that being said, our results did not support our hypothesis that if the dog is younger, then it will have more bacteria present in their gut microbiome. There is not enough data to definitively answer whether the gut microbiome is different between older versus younger dogs. If we were to conduct this experiment again, we would test for different phyla of bacteria to see if there are any further differences between the 2 test groups.